So this week, I'm very pleased to introduce Pieter Cullis, who is from UBC. He was involved with our department at one time, but now, well, I could tell you what his titles are, but they're on his title slide, and he's going to give us a little information about his background. So rather than me garbling his history, let let him do it himself. So Pieter, it's all yours.
Okay, thanks very much, Doug. Yeah, so today, well, the title says it all. Describe some of adventures that I've had over the last, it's coming on for 50 years, of a lapsed physicist and from solid state physics to COVID-19 vaccines. I have a couple of conflicts of interest which are indicated here. I've founded a number of companies, and the ones that are most germane are Precision Nanosystems and Acuitas Therapeutics, as far as this talk is concerned. Okay, I got my PhD from the UBC Physics Department in 1972. It was entitled Electron Paramagnetic Resonance of Phosphorus-Doped Silicon in the Intermediate Impurity Concentration Range. And this was after doing this, I decided that I really had to do something different. It seemed to me that the most interesting problems were outside at least the problems that I was getting interested in were outside the field of physics. Now, Meyer Bloom, who was really one of the more influential profs at that time, had organized a course on biological membranes, and so I got interested in this area and ended up applying for a medical research council. It's now the Canadian Institute for Health Research. For a postdoctoral fellowship, and which I won, which was a surprise to me. It was certainly shaped the direction that I went subsequently. So I ended up in the Biochemistry Department in Oxford in 1973, which was quite a shock because I didn't know the first thing about biochemistry. Didn't even know what a protein was, let alone anything else. I remember walking into the department past the animal room, and that was certainly an unusual environment that you don't encounter in the physics department. Let's put it that way. So anyway, what do you learn as a physicist? One thing is physicists are a pretty tough crowd, and if you leave, you have to leave with honor. And one of the things I took on actually after I'd already got my PhD, and I was doing some learning a little bit about biochemistry, and I decided that I had to write this paper on electron paramagnetic resonance in inhomogeneously broadened systems as a spin temperature approach. And 95 equations later, I came to the end of that. That was published in the Journal of Magnetic Resonance. And so that was really my 1976, the last physics paper that I ever published. But what else do you learn as a physicist? And one of the primary things, of course, is, at least as an experimental physicist, is when you need an instrument that isn't available, you build it. And when I got to Oxford in 1973, late 1973, we had a superconducting magnet, 220 megahertz on the protons, but the problem was we just had the magnet. We didn't have the rest of the the spectrometer built, and so that's what I had to do for the first year or so when I was there, is build this NMR machine that I then subsequently used. We tuned it up to phosphorus as one of the, at least from a biological point of view, as being an interesting nuclei to go after. The other thing you learn is to go after basic problems, and so I got interested in the properties of lipids in biological membranes, and at that time, and actually it remains true today, the basic problems in biological membranes, and this is a Singer-Nicholson model that's still the best model we have of biological membranes. The basic problems were these membranes contain hundreds of different species of lipids, and why are they all there? And we still really have not answered that question. And they have different species. There's lipid asymmetry, so there's different lipids on one side of the membrane respect to the other. So how is that generated, maintained, and what are the functional consequences of that? And so the this kind of boiled down to a couple of areas. One of them is that biological membranes contain lipids that adopt these non-bilayer structures. You can understand why lipids in a biological membrane have to be in a bilayer, because they have to separate an exterior from an interior environment. But why do we have lipids there that on their own will adopt to these non-bilayer structures, such as the hexagonal H2 phase? And then also, what's the influence of ion gradients on the transbilayer distribution? And so these were the problems that we were tackling, quite basic problems. And we're using NMR, phosphorus NMR, to investigate these phase properties. So lipid polymorphism. Of course, with a phospholipid, you have a phosphorus in the head group, and you can use that as a label to delineate the structural properties the structures that these lipids are in, such as a bilayer organization. This is a chemical shift anisotropy giving rise to asymmetric line shape, which changes on going to the hexagonal phase, because you get the rapid movement about these small aqueous channels in the hexagonal phase, causing the line shape indicated here. And if you get isotropic motion, then of course for various structures, you're going to see a narrow line shape. And so it ended up that we could use phosphorus NMR in a productive manner. And we developed what we term the shape hypothesis to explain why it is that various lipids will adopt various structures. And so pretty simple thinking, cylindrical shaped lipids will adopt a bilayer, or the area in the head group region is similar to that subtended by the acyl chains. If the area in the head group is less than that subtended by the acyl chains, then the hexagonal phase is preferred, so we call those cone shape, etc. So this was we proposed that the presence of these lipids in a biological membrane induces a elastic stress, if you want to think of it that way, because there's an intrinsic radius of curvature that you can assign to any group of lipids due to the cone-shaped character of a proportion of those lipids. Of course, they're forced together in the presence of water. Hydrophobic regions will associate with one another, and so you get an increased lateral pressure, which has been suggested to play a role in sealing membrane proteins into the bilayer. It also kind of gives a spring-like quality to the membrane, and when you get two of these membranes that are highly stressed coming next to each other, you can get fusion occurring between those membranes. So in order to investigate the problems that we had to associate with these lipids, we had to develop simple model membrane systems. Biological membranes are just too complicated to sort out the properties of individual components. And so what we wanted was a very simple unilamellar system, as is indicated here, that we could use as a model both for lipid asymmetry and also to look at the effects of ion gradients, etc. And in the early 1980s, there was really no straightforward way to make these small vesicles. And so we built an apparatus to generate these systems. Quite a simple apparatus, but it was very effective. You can just disperse lipid in water. The macroscopic structures micron-sized structures will result from that. So this is a matter of just shaking or vortex mixing. But then if you ram it through a polycarbonate filter with, say, 100 nanometer pore size under high pressure, nitrogen gas about 500 psi, then you'll end up with these unilamellar vesicles coming out the other end. And this, of course, goes back to the physicist approach, which is when you need an instrument, build it. So we built it, and this is a cryo-TEM. This is a 100 nanometer bar down here, showing the sorts of systems that you can generate. And also resulted in a first company that we started, and you'll see that's a bit of a theme. So we invented this extruder device in 1984, and I gave a talk somewhere in New York, and a guy in the audience said he wanted one. So I got a local machinist to make this, and so I got him to make another one for the investigator in New York. But then I got an irate phone call saying, well, it didn't work. Of course, the machinist had no idea what the machine was made for or was intended to be used for. So there was no quality control or anything like that. So me and a few postdocs in the lab formed our first company to market the extruder. And about the only thing we knew about companies at that point was that companies had to have a mission statement. So we developed one. And that was one of the best mission statements that of a company I've ever been part of. Free beer every Friday and parties whenever possible. It turned out that the extruder was a major success, and there's been thousands of these extruders of varying sizes, ranging from the bench size through to $1 million extruders that are used to make clinical grade lipid nanoparticles. And so, there's huge sales that have resulted from that. As I say here, free beer every Friday forever. Let's go back to this one. Anyway, we use these unilamellar systems to ask why these non-bilayer systems are present in membranes. And we showed that if we incorporated increasing amounts of the hexagonal phase or non-lipids on their own, I wanted to adopt these different structures, we could engender fusion between them to form these larger structures. And so this resulted in a model of membrane fusion in biological membranes that's pretty much the one that's generally accepted today. We also use them to ask how lipid asymmetry is generated and what are the consequences. In order to do that, we synthesized an ionizable cationic lipid, and this will be a constant theme actually through this talk. It has a property that it can be a protonated, this is a primary amine here, which can either be protonated or above a certain pH, it will be deprotonated. Anyway, the result of that is that if you have these small vesicles with a more acidic in the middle, say pH 4, about the acidity of a lemon, and pH 7 on the outside, the neutral form, this form here, can very readily move across the membrane. And of course, once it gets to the inside, low pH environment, it'll get protonated, positively charged, and can't get back out. And so you can start to drive lipid asymmetry in these types of systems in a pretty compelling manner. And so this is just showing the kinds of shapes, the shape changes that you can engender in these types of nanoparticles as you move lipids, in this case, from the inside monolayer to the outside monolayer. You can get complete reversions from this kind of shape, kinesite to elongated systems with various protrusions. I'm quite sure that biological membranes use this kind of process for driving transport and other properties. But we got distracted. We got distracted in quite a major way. And we got the way that this happened was that we had to set up these systems with a pH gradient, but in order to measure these pH gradients, we employed a radiolabel version of methylamine, which is a weak base. And so this weak base has a property that can exist again in a protonated or a non-protonated form, but the non-protonated form can move straight across these. The permeability constant it moves very quickly through the lipid environment. And of course, again, if you have a pH gradient, this gets protonated and gets trapped on the inside. And so at equilibrium, you can measure the amount that's on the inside. This is radiolabeled, for example, and that will mirror the proton gradient. So if you have a 3-unit pH gradient, you end up with 1,000 times higher concentration of methylamine on the inside than is on the outside. So what that was thinking a little bit, because a lot of cancer, many drugs, including cancer drugs, are weak bases. And so this indicates a few of them. Doxorubicin is then vincristine. These are all major drugs, major cancer drugs, but they all have this property of being weak bases. primary, secondary, or tertiary amines. And so we asked the question, well, would they respond in the same way as the methylamine? And it turned out that they did. So that we could drive accumulation of these drugs into these lipid nanoparticles, such that we could end up with the concentration of the drug inside over that outside, reflecting the pH gradient across the membrane 1,000 times or more higher concentration on the inside than on the outside. This is kind of a neat technique. That was very robust. And we can, this is just indicated in cartoon form, driving the accumulation of these small molecules into these liposomal or vesicular systems. This just gives you a feel for what you can do. The doxorubicin being a representative anti-cancer drug, we could drive accumulation of doxorubicin into these vesicles. You can see the vesicles clearly outlined here with the kind of railway track exterior, and have these nanocrystals of the drug precipitating on the inside as you exceed the solubility product. So it's a very, as I said, you can get 100,000 molecules or more of a drug such as doxorubicin inside one of these 100 nanometer systems. So we decided to say, okay, well, this is interesting. Maybe we can use these systems for delivery of anti-cancer drugs. And this is a situation that we still have. I can't say that we've beaten the problem yet, but we're well on our way. But right now, if somebody gets, has cancer, for example, and they take the drug is administered systemically, less than 0.001% of these drugs actually go to where they're needed. And of course, they have all kinds of nasty side effects, hair falling out, et cetera, right through to depressing your immune system and a variety of other cardiac problems, et cetera, that can result. So what we need are drug delivery systems to deliver the drug much more specifically to where it's needed. And this is an ongoing effort that we're gradually getting better at. So we started a couple of companies to try to pursue these ideas commercially, starting with a Canadian liposome company in 1987, and then subsequently that morphed into a company called Inex Pharmaceuticals. These efforts were pretty successful. We managed to get 3 drugs approved by the FDA and the European Medical Agency for treatment of disorders such as breast cancer, acute lymphocytic leukemia, et cetera. So we made some real progress here that is now being used in the clinic. And we're on the brink of actually getting that, improving on that substantially. But then in the mid-90s, we decided to say, okay, well, maybe we can try these similar systems, but instead of encapsulating the small molecule drugs, perhaps we can try to encapsulate nucleic acid-based drugs to enable gene therapies. So this really started in the mid-90s. As I said, the CEO that we'd hired said, I can't raise money here putting old drugs in liposomes. You need to be doing gene therapy, which has much more sex appeal. And I guess the investors were demanding that we move in this direction. So I can't say I was that keen about it, because we've been done pretty well with small molecules, but we went headlong into this area no matter. Now, just if you look at Wikipedia for a definition of what gene therapy is, it's a therapeutic delivery of nucleic acid into a patient's cells as a drug to treat disease. So a fairly general process, but the operative point here is the delivery part. The delivery system is absolutely required because nucleic acid polymers are rapidly degraded in biological fluids. if you inject them systemically, they really don't preferentially accumulate where you want them to go. And even if they get there, they can't get into the cell because they can't get across the cell membrane. And so delivery systems are required at every step of the way. Now, gene therapy is really an area of unbelievably intense interest, really because potentially any disease you can approach, you can treat using gene therapy approaches. If you're, I mean, hypothetically, if you can get it to work. This is reflected by, I think it's about 2,500 clinical trials that have been initiated since the early 90s. But this has not resulted in a huge number of successes. There's a few that are based on using a virus, adeno-associated virus, as a delivery system. So most have used that, but there's only been 3 drugs approved. But very recently, and this is what I'm going to describe, particularly for Onpattro and the Pfizer BioNTech COVID-19 vaccine. These are the recent arrivals, and you can see that there's going to be a lot more gene therapies resulting from the lipid nanoparticle approach. So the viral vectors, the reasons why viruses have been so slow to be actually resulting in gene therapies is they have a limited capacity, 5 kilobases of genetic information. They're extremely difficult to engineer. They stimulate immune response. They're very expensive, and they can be toxic, etc. There's been some brilliant work done on these viruses to try to improve them, but I can't say that this is progressing all that quickly. The non-viral vectors have none of these problems potentially, but historically have been relatively poor in their ability to deliver nucleic acid-based drugs into cells, and they can be toxic. And so what I'm going to describe in the next part is really how these issues are being overcome using lipid nanoparticle approaches. Now, the central dogma of biology in terms of the translation of DNA into RNA into proteins is illustrated here. And there's a number of ways that genetic drugs, if you want to call them that, based on RNA, can interfere or augment that process. For example, if we introduce short interfering RNA, siRNA, as indicated here, there are cellular mechanisms that will inhibit very specifically particular mRNAs and actually cause them to be degraded. And so you'll inhibit the translation into the protein, of course, and then inhibit production of the protein itself. Similarly, if you can deliver a messenger RNA, so I got this SI, it should be mRNA, to produce therapeutic proteins, then you can cause these proteins, these mRNAs to be translated and form a therapeutic protein. And so it's these are the kinds of approaches that become possible if you're dealing with nucleic acid-based drugs or for gene therapy. So I'm going to talk about in the next while is the design of these systems for encapsulation of nucleic acid polymers. I'll talk briefly about the Onpattro story, which is a drug that we worked on in the 2005 to 2012 kind of time frame. And then subsequently, I'll just mention a little bit about the COVID-19 vaccine, but it all relies on the same technology. So this is in 95, what we started off doing was saying, okay, we want to deliver these nucleic acid-based drugs into the liver and then get it into hepatocytes. There's a lot of potential targets in hepatocytes that for either silence or express a protein. And so, but you can see this is kind of an ambitious goal. We had to package it, have to then make sure it gets to where you want it to go, and then get out of the vasculature, and then be taken up by a cell, and not just that, but be then after it's taken up, be released into the cytoplasm of the cell. So pretty big challenge. Now, efficient encapsulation of these negatively, obviously the nucleic acid polymers are highly negatively charged. Efficient encapsulation requires the use of cationic or positively charged lipids. So you can see here, just in this cartoon, that you're going to get a hydrophobic entity out of this, and the potential for encapsulation is there. But one problem or one point is that there's really no cationic lipids in nature. They're either net neutral lipids or negatively charged lipids. And so these lipids are highly toxic, and they're toxic for a reason that comes back to lipid polymorphism. That if you introduce a positively charged lipid to a membrane that has a negatively charged lipid in it, you of course get this association, and you'll get wholesale transformation into these non-bilayer structures, which of course is not very good for a cell membrane. And this is the root cause of the toxicity. So we had to find a way around this. And what we did is we went back to the lipid that we synthesized for lipid asymmetry. This is the DODAP lipid, and this lipid is not charged at physiological pH. It's above its pKa. And it's when it gets protonated at pH 4 and is positively charged, but it's not positively charged at pH 7.4 or physiological pH. So it's a much less toxic molecule. So what we attempted to do was to say, okay, well, where it's positively charged, perhaps we can encapsulate the nucleic acid polymers when it's in that state. And if we're really lucky, maybe the whole system will still hold together if we raise the pH. And that's what turned out to happen. We found that we could formulate siRNA and mRNA into these lipid nanoparticles. So we mixed the lipids that were dissolved in ethanol, and you can see here, with nucleic acids that are dissolved in water at a low pH. Do this really rapidly. And one of the first things to fall out of solution is the oligonucleotide surrounded by the positively charged lipids. If you do it fast enough, you can get trapped in its limit size by having other lipids there. So this is a PEG lipid that will fall out of solution a little later and coat these systems and stabilize them. So then we can dialyze away the ethanol and raise the pH. Now we need a rapid mixing process, and so I ended up talking with Carl Hansen in the physics department, and he said, well, you should use microfluidics and microfluidic mixing. I thought, first of all, that meant we were going to get microliters out, but that's really not the case. You can put these things in parallel. And so we developed a technique using lipid dissolved in ethanol in one arm, the aqueous, the siRNA or mRNA for that matter, in the aqueous buffer at pH 4, and we do this about a 3-to-1 ratio on volume, and then dialyze away the residual ethanol, raise the pH when you have your finished system. This turned actually into a company called Precision Nanosystems, which is now the leading provider of bench to GMP, to clinical quantities of material that's now based in Vancouver. So it's turned out to be quite a useful process. But what we have here is really a brand new type of system with a hydrophobic core, without the bilayer track around it that you remember from the liposomal or lipid vesicle systems. It's ideally encapsulated, suited to encapsulation of these negatively charged macromolecules. We can get encapsulation efficiencies of approaching 100%. We can vary the size simply by varying the surface to core lipid ratios, relatively non-toxic, scalable, etc. And so this is still going on in terms of developing more and more sophisticated systems, but this was the system which we then went in and said, okay, are these of any utility in a therapeutic situation? And so this was the cartoon version of the systems that we took into animal studies, and we had a relatively simple model system to look at here. We wanted to silence a gene in hepatocytes, and we had an siRNA that would silence factor 7, one of the clotting proteins. is present in the blood, and it's made in the hepatocytes in the liver. So if we silenced the factor 7 being made in the hepatocytes, then we could terminate the mice after, say, 24 hours of injection, and assay the plasma for factor 7. And it's a reasonably high throughput approach. We could do 2 studies a week, which we did for about 4 or 5 years. We screened over 300 ionizable lipids. This just indicates the progression. They all look pretty similar, but they have very different properties. We found out we found that the potency of these systems was highly sensitive to particular species. And over the course of about a 5-year studies, we went from having to give a dose of 10 milligrams per kilogram of siRNA in a mouse model to be able to silence a gene in hepatocytes, the factor 7 gene, to as little as 5 micrograms of siRNA without increasing the toxicity. If anything, it was decreased. And so we had a huge therapeutic index, therapeutic window to be able to administer these systems. So we can silence the factor 7 gene in hepatocytes. By extension, we can silence any gene in hepatocytes. All we have to do is to engineer the small interfering RNA so that it binds to a different mRNA and causes that to be degraded. And so this is where the clinicians at Alnylam, the company we were working with in Boston, they chose to develop a therapeutic to treat a hereditary disease called transthyretin-induced amyloidosis. Now, transthyretin-induced amyloidosis, transthyretin is made in the hepatocytes in the liver, of course. It's secreted into the circulation, but if there's mutations in the gene, it can often cause formation of fibrils, these amyloid fibrils that circulate around. They deposit pretty much everywhere, but they have particularly nasty ramifications when they deposit in the heart or in nervous tissue. And so following, there's really no effective therapy, and it's a fatal within 5 years of diagnosis. So the multi-systemic disorder, a really nasty one, ranging from CNS disorders, gastrointestinal, neuropathies, cardiac problems. First manifestation is difficulty walking, etc. So a really nasty progression. This is just indicated here for a patient going through this. Pretty nasty because, of course, it's a hereditary disease, and the individuals that might feel the first indication in their 30s probably have a relative who's gone through this and knows what they're in for. Anyways, it was a very simple hypothesis that was you could put forward as much as you have an siRNA to silence transthyretin in the liver, so that's not being produced anymore. You'll reduce the tetramers, prevents the amyloid fibrils being deposited, and perhaps if you're really lucky, you'll get stabilization and recovery from these disorders, particularly if you're able to dissolve the myeloid plaques that have already been formed. So a phase 1 study in human volunteers showed that the dose level of about 1.15 milligrams of siRNA against to silence transthyretin in the liver resulted in a dramatic decrease in the levels of circulating transthyretin, as is indicated here, which would last for a time on the order of 3 weeks to a month. And so on the basis of this, a dose of 0.3 milligrams per kilogram body weight of the siRNA to silence transthyretin was given every 3 weeks in subsequent trials. I'm not going to go through these in any detail. It was an 18-month trial, worldwide trial that was done on this to treat hATTR amyloidosis that was completed in August of 2017. There's over 200 people in that. There was a control arm using sterile saline, And then there was another arm that used the lipid nanoparticles that were containing the siRNA to silence transthyretin. The primary endpoints that were looked at in this study were a neural impairment score. This is neuropathies of either not being able to feel your feet, etc. Patients' perception of their disability, so as the quality of life, overall disability, etc. And so these were the manifestations of the disorder that were followed for these patients over an 18-month period. Now, the results of this trial were absolutely unbelievable, in as much as the primary endpoint, neuropathy improvement, the p-value here, 1 over Avogadro's number, more or less. There's no doubt that this drug actually works. And similarly for the other secondary endpoints. So we've ended up now with a drug that is a stabilizing and potentially curative therapy for a previously fatal disease. So this was a major win, as you can imagine, after 15 years or so of pushing these possibilities forward. So the trademark name is Onpattro, is approved by the FDA in 2018 for treatment of transthyretin-induced amyloidosis. It was the first FDA approval of a siRNA-based gene therapy drug, and it's a pretty big deal. So not only being able to halt the progression of hereditary disease, but reversing accumulated damage. And it starts to really demonstrate the power of gene therapies. So the next is the closing part. I was going to describe the BNT one, or what led to it. It's the BNT162b2. This is the COVID-19 vaccine that Pfizer and BioNTech are now obviously is having some considerable success. First of all, describing the mRNA program, and we're looking here at gene expression in the liver, and then very briefly some of the clinical results. So we asked the question. This is work done in Acuitas Therapeutics, a Vancouver-based company which we founded in about 2009. We asked the question: can we package mRNA in these lipid nanoparticles in the same way that we packaged the siRNA, and if we can silence genes in hepatocytes, well, maybe we can express genes in hepatocytes. That's what was started off in around about 2012. Really a parallel sort of system, but the real difference is, of course, small interfering RNA, just about 13 kilodaltons in weight, it's pretty small. Compare that to, say, a 2 kilobase messenger RNA. I tried to do this to scale, but if I did it, show me the scale, you won't be able to see the siRNA at all. So it's a much larger molecule. So we started to ask the question: does the encapsulation process that has been developed allow efficient encapsulation of the mRNA, and are these LNPs the effective for delivering mRNA to produce protein in hepatocytes? It turned out that the process was very robust, and we're able to encapsulate messenger RNA, as is indicated here. The difference is that whereas we might have 700 or 1,000 siRNAs when we build these systems to contain siRNA, we may only have 2 or 3 of the messenger RNAs which are coding for a particular protein. By cryo-TEM, they look pretty similar to the systems. This is a 100 nanometer bar here to the systems we used for the siRNA. And if you inject these intravenously into a mouse, you can start to, this is using luciferase as the protein that the mRNA is coding for. And we can see expression in the liver, those dependent expressions. So initial indications are that it worked. And this is work that Acuitas has done over the last 8 years to develop more improved systems that are giving rise to increasingly better gene expression levels in the liver for these mRNA-containing systems. And so getting much better at doing something in the liver. But around about 2014, we were approached by Drew Weissman, who is an immunologist who works at the University of Pennsylvania. He'd been working with Catalin Carrico for some years, and they were exploring the potential of mRNA therapeutics as vaccines, and they found ways of modifying the mRNA so they could increase gene expression once they got the mRNA inside a cell. This was basically in vitro, markedly by orders of magnitude. But as Drew said, we have a problem. How do we get mRNA coding for viral proteins, which is what they wanted to do to raise an immune response to the viral proteins, into muscle and immune cells following an intramuscular injection? And so this was instead of going in intravenously as we were for the liver applications, here one's introducing the carrier, lipid nanoparticle carrier, containing the mRNA intramuscularly, getting into, say, muscle cells, and then the viral protein being made, and then exciting an arm of the immune system using MHC1 approaches, as well as getting into antigen-presenting cells and eliciting a different response via the MHC2 receptors. And so this was the investigations that were supported by Acuitas with Drew Weissman. So one of the first things that they looked at was the Zika virus, and so they got mRNA coding for the Zika virus pre-membrane envelope glycoprotein, encapsulated it in the lipid nanoparticle, and then asked the question in a mouse model whether or not these mice were then protected against the Zika virus. And so this was injecting intradermally, and then following up with a challenge at 2 weeks or 20 weeks to see whether a good immune response was elicited. Needless to say, that was completely protective against Zika virus infection at both 2 weeks and 20 weeks. So a very effective vaccine that's probably going to see its way into general use fairly soon. Around about the same time, this is in January or a little bit later, in January of 2020, Acuitas has been working with CureVac, a company based in Germany, and on a rabies vaccine. And so this is the rabies virus. And so the glycoprotein that's associated with that was the mRNA was coding for that glycoprotein. And they announced that in people, this was a clinical study, a phase 1 study, that very, very low levels of messenger RNA encapsulated in the lipid nanoparticle could give rise to a very robust immune response for rabies. And so it was looking very positive as a vaccine. So that triggered a chain reaction. The BioNTech, another partner that Acuitas has been working with, was also working with Pfizer on a flu vaccine. And of course, when the COVID-19 pandemic started in January, February last year, all efforts were switched to making a COVID-19 vaccine. And so here, it's a relatively simple switch. You get the mRNA coding for, in this case, the SARS-CoV-2 spike glycoprotein encapsulated in these lipid nanoparticles, and then see how good a response you can get. And it's obviously pretty good. This is a summary from a press release that Pfizer and BioNTech released in November of last year, and most of you know these results: 95% effective, independent of race, age, gender, etc. and pretty well tolerated. They've indicated they indicated a few months ago that they're going to manufacture up to 1.3 billion doses. Actually, they're up to around about 3 billion now that they project they'll have, that Pfizer and BioNTech will be producing. And there's little doubt that these systems are going to play a major role in ending the COVID-19 pandemic. So this is the efficacy from Israel, a country of about 9 million people. And you can see here they were going through an onset of a second wave around about December. They started vaccinations at this point. They got in front of the line by paying Pfizer and BioNTech a little bit more for their doses, and a very dramatic effect, essentially, as they got to the point where a good proportion of the population was vaccinated. And so this is, a pretty spectacular event. So what I've covered here is, going back to some roots in physics and NMR, et cetera, and then getting more and more involved in some of these therapeutic applications. The applications for gene therapy are certainly quite spectacular. It's been quite a journey. This is not, from the point of view of treating a hereditary disease. And there's many more that'll come along here, as well as the vaccine applications. So what the vaccines are producing is really the tip of the iceberg. You're going to see HIV vaccines, as I mentioned, Zika virus has potential for a universal flu vaccine. Maybe we don't have to change that every year. Malaria and so on. Cancer vaccines, the possibilities for cardiovascular problems, very direct treatments there are becoming apparent, as is Alzheimer's and others. And also rare diseases, inherited diseases, one can see ways of attacking them in a direct manner. One of the points to be emphasized here, that in contrast to our usual way of developing drugs, these systems can be developed very rapidly, because once we know what protein we need to silence or express or edit, we can produce the mRNA or RNA, siRNA or mRNA in a matter of, say, a month or two. The encapsulation is in a day or two. And so you have a new medicine that's highly targeted, that's available on the order of a few weeks. So it's a bit of a switch from the 15-year kind of time frame that we're used to with usual drug development. This has been a huge effort by probably thousands of people, certainly many hundreds. But I want to pay particular tribute to as I mentioned, we started LifeX Biomembranes with a few postdocs in my group. They're still working with me. Mick Hope and Tom Madden, so working for over 40 years, and many others such as Steve Ansel, Yang, Barb, Paulo, etc., who have been working with me for more than 20 years. And so it's been quite a journey with long-term associations. James Taylor, Ewan Ramsey at Precision Nanoceps, and Lloyd Jeffs, who have been involved with the microfluidics approach, Carl in the UBC Physics Department, people at Arbutus, who came up with some of the early ways of developing these lipid nanoparticle systems for silencing genes in the liver. People at Alnylam, who we had a long collaboration with, Marco Cifalini in the chemistry department here, who really broke the problem of the ionizable lipids in terms of making them more potent. These things work in the brain. We've worked with Brian McVicker and others, my own group, and of course Drew Weissman at the University of Pennsylvania, who really was the person who focused us on the vaccine possibilities. So with that, I'll take any questions you may have. I think I'm finishing roughly on time.
Yeah, great. Thanks very much, Peter.